Hello, my name is Greg Friedman. Welcome to my channel where I try to review scientific papers and maybe other sciencey stuff. Today's paper is pretty hard for me. It's outside of my technical domain, but it's very interesting, so I will only review it partially. I will leave the links to this paper in the description down below. Please feel free to download this paper and read it for yourself. You probably should if you need more details. If you like this content, please click the like and subscribe button and don't forget the little bell so we can see each other more often. So let's dive right into this. So today we're looking at paper published in Nature Communications. That's a pretty darn good journal. Hard to publish in. So we're looking at the structure of the plastic-degrading Ideonella and its enzyme bound to a substrate. So we're looking at the structure of an enzyme. Outside of my domain, please forgive me for any mistakes I may make. I'm just simply reading this together with you. This polyethylene terephthalate, PET, plastic is extremely durable, so it sticks around for a while. And in this paper, authors look at this bacteria, Ideonella sakaiensis, and its enzyme PETase. Ase usually means enzyme, and this is an enzyme that consumes PET. So this enzyme converts PET to a substrate, and that substrate hydrolyzes MHETase to the original monomer and ethylene glycol. Fantastic. How's this terephthalate for the environment? Not entirely sure, but that's a topic for another discussion. Bottom line of this paper is there's a possibility that this enzyme can become a valuable resource to advance the enzymatic catalysis of plastic degradation. So we could possibly use bacteria to digest PET. Maybe. Possibly in big quantities, but probably not. But maybe. But let's take a look. Apparently 70 million tons of PET were produced in 2020. Pretty popular. 70 million tons. I'm not sure metric or imperial, but that's a lot any way you look at it. The authors mention that although PET and other synthetic polymer plastics are considered non-toxic, larger particles and microgranules are durable, omnipresent in marine or terrestrial habitats, and accumulate in living organisms. Whoops. Are they toxic or non? You know what? Everything's toxic. Current recycling efforts of PET use very little of it and mostly add huge quantities of virgin polymer. So recycling isn't too terribly effective, suggest the authors, and I agree. There's been some work showing Ideonella can grow on low crystallinity PET films. So these bacteria are able to get their carbon from PET. And this is the reaction. So you have a two-step reaction. PETase breaks down PET polymer into MHET, and then MHETase converts it to ethylene glycol, which is pretty benign, and this TPA, acid, which may or may not be somehow toxic to the environment. I don't know, looks pretty reactive, but I don't know. That's another paper. So this PETase shows higher activity at ambient temperature and on highly crystalline PET. So we need to grind up PET for them to eat it, and we need to run at room temperature, which is pretty nice. Means it doesn't work in a cold though. All plastic-degrading enzymes are known so far display an alpha-beta hydrolase fold. MHETase, however, is likely to possess a scaffold unprecedented for plastic-degrading enzymes. Cool. So these guys, this Ideonella, is a little different than others. Results are very hard for me to interpret. Guys, if you're interested, please download this paper. Link is below. Take a look at this. This is a huge MHETase protein or enzyme that binds this molecule in question and rips it apart. Very cool, complicated stuff. There's interesting effect of mutants on degradation of plastic. Like we're comparing turnover rate to how fast it happens all over the place. This data to me looks very noisy, but these are different mutants degrading plastic. Look at this, catalytic properties of MHETase active site mutants. These are kinetics, and kinetics depend on a particular mutation, but looks like this guy mutates into mutants that have different enzymatic activity, which means if we drive its mutation, we're able to create a diverse community of these bacteria so they can digest different types of plastic, maybe possibly one day. Let's try this. This is an idea for future experiments. And the authors mention while this mutation could be a disadvantage for the bacterium in a natural environment where low substrate concentrations are expected, it is advantageous for biotechnological applications. Yep. So we can mutate this guy to make it eat PET even more. Great! So in conclusion, authors mention that microbial degradation and metabolization of PET and other plastics is a relatively recent thing. Oh, I know. It's hard for bacteria to digest plastic. Plastic is very tough. It's created to be tough, but there are efforts to create new types of bacteria to help us degrade polymers. So in conclusion, I want to applaud the authors for very extensive and poorly reviewed by me work on plastic-degrading bacteria. Rest assured, this work continues by many authors around the world, and even in our lab, we're doing a little bit of work on mutating Ideonella to digest different kinds of plastics. When you visit our lab, we'll show you our morbidostat. It's pretty awesome, and it's degrading bacteria right now. Stay tuned. If you like this, click the like button, subscribe, and the little bell so we can stay in touch more often. Thanks, guys. See you in the next one.